thank you everyone for joining us for today's webinar. I will be talking about some of the best practices if you're going to be working with immunodeficient mice to maintain their health status. So whether you're simply just breeding them or if you're actually going to be using them in experiments. My goal is to share with you some of the considerations that we take into account when working with these mice and give you a toolbox to use if you're going to be working with them on your own. And this is especially with regard to the development of highly immunodeficient mice. So they're fantastic models, but they're super sensitive to traditional pathogens, opportunists, and organisms that are not typically pathogenic in mice. And so, how does that affect how you care for them and how you breed them and what their husbandry is? So this presentation is going to be more focused on the practical side of working with these mice, and I hope that it'll be really valuable to you. Before we do that, however, we always like to give a little bit of introduction and background about the Jackson Laboratory for those of you who are unfamiliar with who we are and what we do. So the Jackson Laboratory, we're an independent nonprofit organization that focuses on mammalian genetics research to advance human health. Our mission is to discover precise genomic solutions for disease and to empower the global biomedical community in the shared quest to improve human health. And we advance that mission in 3 different ways. The first is through performing primary research. We have our own research staff with their own private funding means, such as NIH grants, that investigate the genetics and biology of human disease, partly using the laboratory mouse as a model. We also provide scientific services and resources to investigators around the world. So in addition to our clinical and research services that some of you may already be familiar with, we also have some freely available resources that I'll briefly touch upon in a little bit. And lastly, at the Jackson Laboratory, we're very committed to education outreach. So in addition to webinars, such as this one in which you're participating in right now, we also sponsor traditional conferences and hands-on workshops, as well as education of students through all ages through courses, internships, and other programs, all the way from high school internships to postdoctoral fellowships. The Jackson Laboratory is the world's largest repository for mouse models of disease. We are NIH funded to import and cryopreserve as many mouse strains as possible to ensure their availability for all time. So among the more than over 8,000 strains that we distribute from both of our, more than 10,000 strains now, it keeps on growing and growing, we distribute from both our Bar Harbor and Sacramento facilities. About 1,500 are maintained as live colonies. The rest are cryopreserved in our facility. Jack's mice are the best characterized and referenced mice in published research, and much of this can be attributed to rigorous animal health standards and unsurpassed genetic quality, which is a topic that we'll be covering in today's talk. So in relation to producing the most reliable, reproducible data, obviously health status is going to be one of those factors, and if you're working with immunodeficient mice, health status is key. So what many investigators may not realize about the Jackson Laboratory is that it's through the sale and distribution of our well-characterized and heavily referenced common inbred strains, such as the Black 6 or Balb C mice, that supports the development, the collection, and distribution of all those specialty mutant mouse models, as well as providing funding for some of our more freely available resources. And very briefly, here are those resources I'd like to just point out to you in case you haven't accessed them already. The first is our Jack's Mice database, and this is essentially our inventory, our catalog of the strains that we have available that you can access through our repository. Each data sheet for each strain actually has a lot of other very useful, helpful information, not just about the phenotypes and the genetics associated with the strain, but also about the husbandry, genotyping resources, breeding considerations, things like that. MGI is a database that is curated by a curation team here at the Jackson Laboratory, and it is a fantastic resource, the one-stop shop for all things mouse genome related, mouse gene related. And also, if you're looking for a strain and you have a particular mutation in mind or particular gene in mind, MGI is a great place to go because it'll link out to give you information about where that strain is available, if it's at the Jackson Laboratory or if it's available somewhere else publicly. And I want to leave these other two on the mouse genome database and the other databases. Those are live links, so when you receive the PDF of this presentation, you'll be able to access those right away and take a look. All of these are very fantastic, helpful resources for anyone who's using mice in research. Ooh, a little fast there. Okay, so let's go ahead and give you a little overview of what I'd like to cover today. So when it comes to housing immunodeficient mice, caring for them, and then using them in experiments so that you get really high quality, reproducible data, three components really will come to play when you're considering the care of these mice. And these three different components are represented here on this pyramid on this slide. The first is obviously the mice. They're the functional unit of the breeding colony, or functional unit of your experiments, the key the key host in some cases, or it could be the model that you're working with. Obviously, the biology that's inherent in that mouse, what mutations do it carry, what transgenes, what kind of phenotypes do they develop. This is all very important to understand so that you know the best way to handle them. How do they breed? What kind of support do they need? What kind of care? What kind of veterinary attention? So make sure that you understand the basic functioning working unit of your colony. Just because it's a white mouse doesn't necessarily mean that you understand what it is. Take the time and really, learn more about it. So we'll talk more about what the different immunodeficient mice are, the more common ones that are used in experimentation. Obviously, we'll be talking about the NSG, but we'll talk about others as well. The second functional unit here is the people. So, the people, anyone who comes in contact with the mouse, it could be the researcher, it could be the caretaker, it could be the veterinary staff, anyone who has some kind of interaction with that mouse or plays a role in how that mouse is cared for in your facility. Cage washers, technicians, laboratory staff, anyone who might be within that realm of that mouse room has an effect on the mouse. And how are they going to be interacting? What kind of precautions are in place to protect them or to prevent the transmission of potential mouse antigens or opportunists, pathogens or opportunists into that mouse? So how are they? What kind of regulations or what kind of considerations should you have in place? We're talking about the people that are there. And lastly is the environment. So obviously the habitat, if you will, for where that mouse is living is going to have a profound effect on what the health of that mouse is and what kind of experimental data you're going to be getting out of it. Where the cage changes occur? What is the cleanliness level? How often are are pathogen tests done? What kind of sentinels are you using? All of these are the effects all these are considerations that may affect what kind of data you're going to be getting out of that mouse. And so we're going to be talking about all three of these different factors in today's talk. So I hope you'll be able to glean something that'll be helpful as you perhaps start to plan whatever experiments that you're working with, or if you're trying to troubleshoot a current a consideration, if you have a trouble in your current colony. So I have three main learning goals that I hope you walk away from this presentation with. The first one is I will introduce a variety of different mouse strains. In particular, I'll be discussing traditional immunodeficient strains, but I'll also point out that some commonly used standard inbred strains have inherent immune deficiencies, and so it might be useful to implement some of the suggestions that I'll discuss for handling for the more traditional models into your handling protocols when working with those mice too. We'll talk about those different immune deficiencies. We'll talk about common types and signs of common infections, specifically in the non-scid gamma, the NSG mouse, and how what should you look for, and how do you mitigate? What's the best course of action? How do you keep that mouse from, keep that infection isolated? How do you keep it from not spreading through your colony? And lastly, I'll be giving some pretty specific descriptions of the processes and caging recommendations for the successful care of NSG mice. And usually anything that will apply to NSGs will apply to the other immunodeficients, but we'll talk about some differences between the immunodeficient and immunocompromised mice as we go along. Fantastic. Okay, let's go ahead and start right on in with a little review of what immunodeficiency in mouse colonies are. What are those strains that we're talking about? So the general strategy for the development of immunodeficient mice for research has been to ablate more and more of the animal's immune system with each new model to support better engraftment of human or mouse tissues. This slide provides a very high-level review of the major categories of immune cells that constitute mouse and human immune system. And I want to provide this just because I'll be referring back to it when we talk about individual strains, and so this will be a quick way to show you what the immunodeficiencies are in each one of those models. So as by way of a quick review, there are two main branches of immunity in mice and in humans, actually in many mammals. The innate branch of the immune system can be considered a first-line defense against pathogens and foreign invaders. It contains various components that are hardwired to recognize and respond to diverse pathogens. Innate immunity includes the complement system, which is derived in lysing pathogenic bacteria and cells bound by antibodies. Macrophages engulf dead and dying cells. Granulocytes participate in allergic and inflammatory responses. And NK cells, or natural killer cells, are a population of cytotoxic cells that can identify and kill pathogen-infected cells and tissues. So it's the adaptive, that's the innate immunity side. And the adaptive branch of the immunity is probably maybe more familiar to you if you have a small background in immunity. The adaptive branch of the immune system includes B cells that produce antibodies and T cells that synergize with those B cells to elicit a specific immune response. So really, if you think about any kind of vaccine development, you want to produce a functional antibody, this is the adaptive immunity, this is the side that we're talking about. So also a part of the adaptive immune system are dendritic cells. Dendritic cells are the antigen-producing cells that get that whole ball rolling by presenting immunogenic peptides to the T cells. And then up above are cytokines. So cytokines, we put them kind of as this bridge between the two different branches. These are more commonly known as interleukins. These are the secreted proteins that regulate differentiation of immune cells, as well as communication among different cell populations. They regulate maturation, signaling cascades between different cell types. So these are really the communication of the immune system. So in the first part of today's presentation, I'm going to go through some various common immune deficient mice that JAX distributes, and I'll come back to this slide and this kind of schematic to point out which specific components of the immune system are knocked out, and how these deficiencies allow the mice to engraft more efficiently various human-derived cells and tissues. I like to think of immunocompromised strains as fragile superheroes. So essentially, the more severely immunocompromised and the ones that we traditionally acknowledge as immune deficient mice, they're weak. They have some of their defenses taken away from them, and it's because they're weakened and makes them such powerful models or such powerful hosts to be able to accept xenograft tissues. And some of these are listed on this slide. Some of the more common traditional ones that I'll be talking about. We'll start at the top. We'll start talking about the Nudes, then we'll talk about RAG1, RAG2 deficients, quick review of SCIDs, and then we'll focus a little bit more highly on the NSG, the more highly immunodeficient model. So let's talk first about Nudes. Nude mice, obviously so called because they are nude, they're hairless. Nude mice have impaired adaptive immunity because they lack T lymphocytes. The Nude mutation is a mutation in the FOXN1 transcription factor, which is involved in not only hair follicle patterning, but also in the development of the thymus. So mice that are homozygous for that FOXN1 Nude mutation are athymic, which means that there's no place for the T lymphocytes to go and mature. So they have no mature functional T lymphocytes. And because you can see the color is kind of drained out of this T lymphocyte bubble to represent that. And they also have a slight defect in the B lymphocyte, and that's because really for adaptive immunity to properly function, B and T lymphocytes need to synergize, need to work together. Nudes, however, you can see that they really retain the whole side of innate immunity. So the take-home message for nudes is that essentially they're good for something aggressive, like a cancer cell line, some kind of immortalized cell line, something usually adherent. They're bad, if not terrible, for engrafting primary human cells like stem cells, because there's only this one chink in the T lymphocytes. There's not really any other immunodeficiency to speak of. The next one we'll quickly review is the RAG1 mutations. And actually, we're going to talk about RAGs and the SCID mutations in kind of the same vein, because even though they're two different mutations, they represent two different parts of this pathway that lead to the absence of mature T and B cells. The advantage here is with these particular ones is that they're on a congenic background. These are on congenic C57 Black 6 background. Which is great if you want to do transplantation or adaptive transfer experiments, but it's not a great host for primary human cells because, again, much like the nudes that we just discussed, they retain this active functional innate immunity arm. Scid mice, as I already said, have a similar phenotype to RAG1, RAG2 mutants. Again, they are two different sides of the same pathway. RAG1 recombinase is the enzyme that mediates the genomic rearrangements that allow for the expression, the creation of specific T and B cell receptors. And the scid mutation is it was a spontaneously occurring mutation in the PRKDC gene, which is involved in DNA repair. So with that, if you don't have a RAG1 recombinase, you can't rearrange the genome to create B and T cell receptors. And if you can't repair the damage, in the case of the scid mutation, the damage occurs, but then the cell will recognize that as damaged DNA, and the cells will apoptose. So you can't create the damage and you can't repair it. Because both lead to defects in mature T and B cell lymphocytes in the RAG and scid mutations. Moving on, when the scid mutation was introduced into a Nude background, it created a very commonly used model on that, which is a Nude scid. So the Nude background, if you combine, let me go back here, because I want to show you one more time. The B6 scid brought defects in T and B cell lymphocytes on the adaptive side of the immune system. But if you take that scid mutation and cross it into a Nude background, create a congenic Nude scid, now you add that defect to T and B cell lymphocytes on the adaptive side to a phenotype conferred by the non-obese diabetic background, which is some deficits in innate immunity. So the Nude background contributes reduced innate immunity in the form of no complement, reduced macrophage activity, and reduced NK cell activity, although they're still there. So when you start combining these two together, now you have chinks in the armor of the immune system on both sides. Now these Nude skids represented a model where you can start to begin to put in or to engraft primary type tissues, not just cell lines or derived more aggressive type materials, but now primary engraftments. Nude skids have been a great host for leukemia and lymphoma cells, particularly those from cell lines, but also primary ones as well. They can also support some humanized models, some stem cell engraftments, although with variable efficiency. But they did represent a great step forward in the immunodeficient hosts. NSG mice, this is the next step, is one of the most immunodeficient strains that currently exists. The big difference here compared to the Nude scid is now we have a full absence, a full knockout of natural killer cell activity, and you can see cytokines are now dimmed out in this picture as well. And that's due to the addition of the gamma mutation, which is the IL-2 receptor gamma, which is part of, it's a common receptor component for a number of different interleukin receptors, and so without that, you don't have proper cytokine signaling, and you don't have a natural killer cell development and maturation. So obviously, this is the NK cells were the greatest impediment they are the greatest impediment to human tumor xenografts and humanization of the mouse immune system by engraftment of human hematopoietic stem cells. And so the NSGs, or Nude scid gammas, really represent a great leap forward to finally being able to efficiently engraft primary human tissues. So I'm just, this is all that I just reviewed, but again, I just want to highlight again the features and benefits of this NSG. It carries a number, so many chinks in the armor of the immune system, many immunological deficiencies. The Nude background confers lots of innate immune deficiencies. The scid mutation prevents development of mature T and B cells. And then when you add on the IL-2 receptor gamma gene knockout, now you have reduced cytokine signaling and blocking of NK cell development. Now, the benefits of these compared to other previous immunodeficient strains is, first of all, there's no scid leakiness. In a B6 scid, after a certain amount of time, as a mouse ages, you'll begin to see some mature B and T cells develop in that mouse. With the NSG, that's not the case. It's a long-term, stable suppression. There's no leakiness of that scid mutation. The NSGs also have a longer lifespan than Nude skids. Nude skids develop a thymic lymphoma somewhere around 7 to 9 months of age. Nude skids don't develop that thymic lymphoma, and they have a pretty long lifespan, lifespan around 22 months. So if you're doing more long-term engraftments, more long-term modeling, this could be very useful for you. We already talked about the thymic lymphoma development, so this is also, you're going to have a reduced amount of confounding lymphomas if you're doing engraftments. So the NSG, because they are so highly immunodeficient, they really have revolutionized research in many areas, including everything on this list. We've already talked about. I've mentioned some of these: primary tumor engraftment, human hematopoiesis, humanized mice, infectious disease, and regenerative medicine. And it's because of that they've been such a revolutionary model, because they allow the engraftment or the incorporation of xenograft tissues from a primary source very, very easily. So let's go ahead and talk more about the specific considerations for carrying and housing of NSGs. So what we'd like to move in now is to discuss some of the infectious disease concerns that come up when working with NSG mice. So being the most immunodeficient of all mice that I've discussed so far, they're extremely sensitive to infection by a broad range of pathogenic as well as opportunistic microorganisms. They're also sensitive to infection by microorganisms that are not necessarily considered opportunistic pathogens in immune-competent mice. So few reports on the sensitivity of NSG to infection by such pathogens, and there are not many reports of this in the literature, but we do receive occasional reports from investigators using NSG where they see high mortality in their colonies. There is one publication in particular that I want to draw your attention to, which is down at the bottom of this slide here. This was published by Jack's investigators, in which we discuss in greater detail the sensitivity of NSG mice to infection by opportunistic and commensal organisms. Because we get these reports, we have to rely on people's experience and what we're finding. And so what people have told us, and what some investigations have found, is that the NSGs, because they have a higher level of immunodeficiency, they have more sensitivity to things that would not normally cause disease in other immunocompromised mice or immunocompetent mice. So things like pathogenic microorganisms, obviously agents that could cause infectious disease in immunocompetent hosts are of concern for NSGs. Some opportunistic microorganisms that immunocompetent mice can, really fend off well, can be a problem for NSGs. And even some commensals, some of those microorganisms that commonly reside within normal host tissues without causing disease, can be a problem for NSG mice if there's a route or a wound, an open abscess, something that will aggravate that, and then those commensals can colonize a different location. So NSGs have some particular considerations to keep in mind, that they're going to be more sensitive to microorganisms and to pathogens that may not have an effect even in other immunocompromised strains. So what are some of these pathogenic and commensal microbes that are a threat to NSGs? So this slide has a list, and it's obviously not exhausted, but this slide lists the sorts of microorganisms that we have cultured from NSG mice. The list includes some of the usual suspects that we might have expected to find in such a list, including Pseudomonas, Pneumocystis, Staph aureus. Surprisingly, the most common bacterial infection that we see in NSG involve Enterococcus, which is usually a commensal in mice. It's part of the animal's normal gut flora. In severely immune deficient mice like NSG, components of that enteric gut flora can colonize open wounds, like the more commonly associated opportunistic organisms like Klebsiella do, and then they develop a clinical infection. So in a little bit, I'll talk about some of the steps you can take to try and safeguard NSGs and other immune compromise from these types of infections. So for some of these opportunistic infections, like I just said, it's really the main concern is that microorganisms that are normally present in the gut flora of healthy mice can become pathogenic in NSGs. And the clinical signs of this is something like a hunched posture, a scruffy coat, and also we see a higher incidence of this in females, twice as likely to be affected. Often this is because the females, if they're mothers, if they have had a litter recently, there's more of a chance for stress within the vaginal opening, or if she's nursing, then the lactation and nursing may lead to introduction of those commensals into tissues where they shouldn't normally be. So let's talk about some of the contributing factors. So to help prevent vascular spread, some of the contributing factors to mortality in a research setting. So some of the opportunistic factors can take these other kinds of routes. They can manifest in urinary tract infections. This is very common. This occurs from normal intestinal flora colonizing the urinary tract. If if your NSGs are getting some kind of estrogen supplementation, it can increase the risk of that. So if you have a breast cancer tumor or a cell line, you're doing some estrogen supplementation, that can be some of the other a risk factor if you are doing the supplements. They may be associated with ascending renal infection and pathology, depending on the severity of the UTI and how long it goes before diagnosis. These infections can also be part of a secondary infection after a primary insult. So anytime the barrier of the skin is broken, if the mice have been fighting, there may be a risk that the skin wounds can be colonized. Or if you have some manipulation of the mouse, if you're doing recurring injections, IP injections, anything with has a needle breaking the skin barrier, that's a potential risk that you can have a secondary infection occur from the colonization of that site by commensal microbes. There's also some oral routes as well that have been reported. And I've already talked that breeding, often lactating females are more susceptible, again, because the lactation and nursing can aggravate the tissue and create a site that the commensals can colonize. So in the next couple of slides, I want to talk about some of the common lesions in the infections that we observed in the immunocompromised mice and in NSGs, and NSGs in particular. So these include, first of all, infected skin wounds and cellulitis. These are lesions from which we can culture microorganisms and that are secondary to the prior insult, such as everything that was in the previous slide, a bite wound, a needle stick, so a nursing female, anything that has that resulted from a broken skin that the commensals managed to colonize. They can also present with abscesses either within the skin or internal organs. Otitis media, which is inner ear infections, and infections involving the eyes, such as conjunctivitis, and panophthalmitis, which is inflammation of the inner structures of the eyes. So again, anything that has an insult, if there's aggravation or scratching, then those commensals can get into the eye tissue. Or they can present with localized and widespread infections involving internal organs, liver, heart, lungs, uterus, sex glands, etc. The most common infections, again, are the urinary tract infections that come in a variety of forms. The UTIs can come in cystitis, which is inflammation of the bladder, kidney infections, or the ascending pathology that I already mentioned. UTIs, again, are diagnosed in female NSGs almost twice as often as in the males. So the organisms that typically infect NSG include the normal suspects of traditional pathogens and opportunists, but again, also the commensal organisms. So it's difficult to generalize about the conditions that contribute to these infections in NSG mice because we've observed them in NSG mice in nearly all barrier levels in our mouse facilities, from worms with loose barrier conditions to those in their highest barrier levels. Problems are most often seen in mice that have been in the facility for a while. So these are mice that, they're older mice, they've been exposed to a bit more over time. And infections typically are sporadic and don't affect the majority of mice in the colony or even all mice within a cage. Typically, we find infected individuals within a cage of healthy animals. And so the mindset isn't so much that this is going to be a widespread epidemic or a widespread pathogen that's going to go rampant throughout your mouse colony. However, again, being able to look and have your clinical worker, have your vet staff, if you find a sick mouse, be able to make sure that you look at the other mice in that cage because they may not be affected. Again, lots of these infections depend upon some kind of skin break, some kind of barrier breach from that, some fighting or needle sticks, something like that. And so making sure that going in with the mindset that this may likely be an isolated case may help with may likely affect the way that you mitigate and your strategies are to prevent it from happening in the future. So I just want to point out that there another health concern for NSGs is not just from the pathogens and potential infections, but there are also, if you have a colony, especially with older mice, you may be seeing some spontaneous tumors in NSG mice, and this has been reported, and we've noticed it ourselves. Even though the incidence of thymic lymphoma is greatly reduced, in NSGs compared to like the non-skids, they do occur. It's a very low, very rare occurrence, again, happening in older mice. Otherwise, they do present, the older mice present with some common tumors. Osteosarcomas and mammary adenocarcinomas are also very common to find in these mice as they age. If you want some more details and the numbers of that, these two publications are very helpful to understand kind of the rates of spontaneous tumors in NSGs. So now that we've kind of reviewed what the overall infectious disease, infectious agents are in NSGs and what the overall health concerns are, let's go ahead and describe the husbandry and caging recommendations for successful care of NSG mice, so that you can have more strategies to mitigate, and how do you care for them, and what are your strategies are to mitigate that risk. So I do want to talk about how clean is clean enough. Obviously these mice are immunodeficient, and you want to house them in a clean environment and something that limits their exposure to microorganisms. But how clean do you have to get? So there are several strategies that are used to decontaminate objects that could potentially carry microbial pathogens, including sanitization, disinfection, and sterilization. So sanitization, which provides the lowest level of decontamination, reduces the amount of microbial organisms in an inanimate object. Well, I could say that properly. Disinfection, which is the next step up, is the next step in limiting the amount of the decontamination achieved. So disinfection destroys or inactivates most microbial organisms on inanimate objects. Finally, sterilization is the complete destruction or inactivation of all microbial organisms on a surface. So there are many methods used to decontaminate objects, including chemical and physical means, so like heat, steam, filtration, radiation. And the method you choose will be dictated by the object, so how can it actually go through that sterilization process and survive. And in the case of immunodeficient mice, the level of immunodeficiency of the mice being housed in that room. So when you're working with NSG, something that's so immunodeficient, we highly recommend sterilization. Do everything you can to completely destruct or inactivate those microbial organisms on anything that's going to be coming into contact or in the presence of that animal. So, so in terms of their housing, how clean is clean enough? Excuse me a second. Being highly sensitive to infection by such a wide range of organisms, there are some extra steps that you should be mindful of when working with them to help minimize their exposure and ensure their health status for the NSGs. So some of these, first of all, barrier practices are highly recommended. However, barrier practices adequate to maintain nude or even scid mice may not be adequate for NSGs. To that end, we recommend working with NSGs, sanitizing hands before gloving, so wash your hands, disinfect surfaces, laminar flow hoods, all the surfaces, experimental equipment, the floors, the walls on a regular basis, sterilizing your tools, anything that the mouse is going to be coming in contact with, so tools, forceps, scissors, ear punches, bedding, food, cages should all be sterilized. We also recommend more frequent cage changes. The longer that the mice, sit and as the mice exist in a cage, they're going to be obviously, eating, creating, fecal matter, and the longer that they reside in that situation, the more likely it is that they're going to pick up a commensal if they have an open wound or something like that. So we recommend more frequent cage changes to constantly kind of clear their environment of whatever pathogens may be shed into their cages. Highly recommend monitoring for pathogens frequently. We do opportunistic pathogen testing by fecal, colony or dirty bedding or sentinel testing, also recommended. And doing all your work under a HEPA ventilated hood. So the hoods will regulate or create that airflow that will minimize spread of particulates and other micro-size droplets and things that might spread germs from surface to surface. So doing all your work under a HEPA hood will prevent that exposure to your mice. In terms of the personal protective equipment, our PPE, we we recommend using sterile scrubs, gloves, and dedicated shoes and a shoe cover. Every time you come into the room, these should be put on de novo, brand new and clean. Using face shields and hair or beard bonnet and mask and goggles to really limit the amount of movement from the airflow from your breath or any particulates that might drop from your hair onto anything that the mice come into contact with. Putting on a sterile smock, again, another layer to prevent those particulates from being shared. Or, and if you're able to do it, we do the air respirators, the PAPRs, powered air purifying respirators. Anything you can do to minimize the amount of, again, particulate or airborne matter that might be shared between, or from mouse to mouse, from cage to cage, or anything that comes from you that can come in contact with the mouse, because you don't want to introduce any microorganisms from the commensals on your own skin into these mice. So we also have some suggestions that that might help to create a specific dedicated area that is clean for your NSGs. We highly recommend, at least if you're unable to do something like a shower or a clean peel into the room, make sure that you can do what you can to have a clearly marked, clearly delineated entry room or space. So either it's an anteroom, if there's kind of like a pre-entry room before you get into the mouse room, or a dedicated hallway. Limit the amount of people who have access to it. Clearly mark it with tape or signage. Even put tape on the floor. Anything you can do to help delineate that. Even just, like I said, some tape on the floor can help limit the amount of traffic into there, and amount of shoes and feet that can carry particulates that can bear some microorganisms in. Limit your entry. Make sure that only people who go into that mouse room are the ones who absolutely need it, and to know what the traffic is like. Not going in and out all day. Maybe not a great idea. If you could limit your time in there to one time in, one time out, that really just reduces the amount of airflow. It reduces the amount of particulates that have the chance to come in. And lastly, air shower if available. We used to do wet showers, but in our facility we use air showers now. They are more time efficient. They blow particulates into a HEPA filter, and they're a little more user friendly. It takes about 30 seconds to a minute to do the air shower in, which is what the gentleman over here is doing as he goes into that mouse room. Again, it's all about limiting any particulates that might be spread by airflow that might expose your mice to pathogens. So when it comes to environmental conditions and the husbandry of mice, there, that old adage, an ounce of prevention is worth like a pound of cure, something like that. Really, anything you can do to limit the exposure is recommended. So I'll say again, sterilize or disinfect anything that may come into contact with the mice. And sterilize or disinfect means you do something like autoclaving. We autoclave our boxes, we autoclave the bedding, the water bottles, lids, everything is that we can autoclave is autoclaved. You can also use things like ethylene dioxide or vaporized hydrogen peroxide if you need to do a surface disinfection. Use laminar flow hoods or biosafety cabinets when working with human pathogens whenever possible. Again, laminar flow hoods with filters on them will prevent that airflow. Do any manipulation of the mouse, any cage changes under those hoods, and you'll limit the amount of exposure for your mice. Use micro-isolators or individually ventilated cages, IVCs. In our facility, we use the individually ventilated cages. Micro-isolators can be useful if you have if your animal room space is limited and you need to house these like NSGs in a room with other mice. Then the micro-isolators can be really helpful for that, and make sure that they do have the HEPA filters. Cage changes again, always changed in a sterilized laminar flow hood. Do them more frequently. Disinfect your gloved hands anytime or anything that goes into the hood. And whoever's manipulating the mice, changing the cage, should disinfect everything when it goes into the hood. And anytime their hands leave the hood, their hands need to be re-sterilized to go back in. Super aseptic technique. Disinfect everything between cages. Wipe everything down again. If you're using forceps, do a Wescodine dip, a disinfection between cages. And again, anytime your hands leave that hood, disinfect them again before you put them in. Do due diligence to make sure that you're not spreading anything potentially to your mice. That's the purpose of the laminar flow hood. And if you follow those steps, the risk of transferring anything, any particulates, is greatly reduced. So in terms of the barrier conditions that we use at the Jackson Laboratory, all of this, pretty much everything that I've just described is what we do. Our barrier conditions and the maximum barrier for the NSGs, sterilized individually ventilated caging, sterilized feed and water, sterilized disinfection, or disinfection of all supplies and equipment. We do air shower entries. We change into clean room process scrubs, smock, and shoes every day, so those are washed. And then every time you change into, it's a new set. Gloves, air hat or mask, cap, shroud, and face shield. Again, really limiting any particulates or particles that might come from breathing or from your eyes or hair or facial hair or anything. And then under these conditions, what we found, under these max barrier conditions at Jackson, bacterial disease in the NSGs is extremely uncommon. We really don't see regular or even any noticeable levels of bacterial disease in NSGs in our colonies. So anytime that there's a risk, anytime that you're doing any manipulation with the mice, which may be an experimental procedure, or if you're just moving it from moving the mouse from room to room, if your surgical suite is down the hall, something like that, consider that as an opportunity to do everything you can to protect that mouse, because obviously it's a vulnerable time if you have to move that mouse or manipulate it in any way. So if you do need to, do any procedures with your mice or transport them, again, follow the same kinds of considerations. Take the time to disinfect any equipment or the environment, especially if it's a shared equipment, shared room. Just assume if it's shared, assume that it's dirty and clean it again. It's really that was my mantra when I was doing lots of engraftments into immunodeficient mice. So anytime I use a shared equipment, I was in there scrubbing everything down again and disinfecting. So do it, because you never know how diligent the person who used it before you was. So do it again. Sterilize all of your smaller tools by autoclaving. So autoclave all of your surgical equipment, autoclave your tips, whatever it is you need to do. Anytime you're going to be doing engraftments of any tissues, again, it's a potential source of contamination or potential source of pathogens for your mice. So if you're going to be engrafting a cell line, you get a cell line from a colleague. Do MAP testing, do the mouse antibody production testing on it, and make sure that it's going to be safe and it's not carrying mouse pathogens before you engraft it. Lots of institutions have regulations about this, and it should be done, but check and see if it's part of it in your facility. Also, when you do any movements with your mice, if you need to take them down the hall, you need to take them to a different room, use secondary containment during transplantation. So put the mice, put the boxes in a big plastic bag and sterilize it before you put it into the hood where you get to. Use a sterile smock, something as a secondary containment during the transportation, just to minimize exposure during that very vulnerable time for your mice. Do want to quickly touch upon, if you're going to be maintaining NSG mice, likely that you're going to be breeding them. Just want to give you an idea of what the breeding is like for these mice. In terms of the genotypes, the SCID mutation is homozygous in both males and females. The gamma mutation is the IL-2 receptor gamma gene is on the X chromosome, so females can be homozygous and males are hemizygous. If you breed home females with hemi males, all the progeny is those are obligate genotypes for the home females and hemi males. If you genotype your breeders, you confirm the genotype of your breeders. You don't need to genotype your pups, again, because they're an obligate genotype. We replace breeders every 8 to 9 months. They're quite good breeders with a pretty long lifespan. So if you want to err on the side, maybe even every 7 to 9 months. If you're not getting any litters, or if the mice appear sick, go ahead and just replace those breeders. In our facility, we follow a 120-day non-productive before we replace them. But otherwise, if you have a concern, go ahead and get a new breeder in, so you're not wasting the time. And we also, our colonists and other users of the strain have reported that the NSGs have, a bit of a gentler disposition than non-scid mice, and they weigh slightly more. They're slightly bigger mice than the non-skids. In terms of care and husbandry suggestions for the NSG, the food, again, sterilize it. It comes in contact with your mice, obviously. Sterilize it. It could be filter sterilized or autoclaved. It should be filter sterilized and then autoclaved or irradiated. In our facility, we acidify our water from using hydrochloric acid. We acidify to 2.5 to 3 of a pH to control Pseudomonas species, and we autoclave it. In JAX, we do not add prophylactic antibiotics to our water. We'll talk more about prophylactic antibiotic in a bit, but we don't do it. We don't recommend it. Although each facility, each institution may make a different judgment call on that. We also monitor pathogens regularly. So we monitor pathogens in a number of ways. We do direct pathogen monitoring from if we find an infected mouse in our colony, we culture from that mouse. We'll do indirect monitoring, which is through sentinel mice. And we also do routine fecal monitoring for opportunistic pathogens from our colonies as well. So it's a very common frequent testing schedule, so that we're staying on top of any pathogens that may arise in our colonies. So for pathogen monitoring, for the indirect, we do sentinel mice. So sentinel mice are those that are used to detecting the pathogens present in the room, and so there's a number of different kinds of sentinel mice. They can include dirty bedding, which is just taking a bit of that bedding and putting them in with the mice. And this is mostly for fecal-oral type microorganisms. We do cage contact sentinels, and this is for direct contact, anything that's passed through direct contact aerosols, urine, or again, fecal-oral. We can also do exhaust air sentinels. So this is for anything that might be aerosolized and shared, coming within that rack or something within that caging system. We use both immunocompetent and immunodeficient mice, which includes NSGs periodically as sentinels. And obviously being able to use both immunocompetent and immunodeficient mice gives you more information about what kind of pathogens you're dealing with when you do that. So for the health monitoring considerations, a couple different factors go into our health monitoring program for immunodeficient mice. So when working with immunodeficient mice, it can be beneficial to expand your health monitoring surveillance to detect all microorganisms that you want to exclude from the barrier, including the opportunists. So one thing here, I do point here that shedding is often intermittent. So keep in mind that shedding of these organisms from infected mice may be intermittent, so increasing the frequency with which you screen mice via non-lethal sampling, like fecal samples or oropharyngeal swabbing, may improve your detection of them. So something that's going to be non-invasive, a little bit more easy to do it more frequently in terms of the monitoring, which will give you a better idea of what might be present. At JAX, we monitor the health or status of our colonies using colony controls, primarily as our sentinels. So colony controls are mice taken from our actual colonies and then grouped housed for a period of time, after which they're sacrificed and then tested for pathogens. So it's a good way to get a bigger snapshot of what's happening within the colony. Understand that testing may fail due to transient nature of some infections, so the best strategy there is to increase the frequency of testing. And then for anything that we...
Oh, lost connection. Is it coming back?
My apologies, everyone had a little internet flicker there. I hope you can all hear me again loud and clear. My apologies, a little technical difficulty. So doing direct tests and not serology for severely immunodeficient mice is also very helpful, as sometimes the infection may not present with serology or with any kind of measurements that you can take from that. You have to do a direct test for it. So anytime you're interested in looking at what our health reports are and what our health testing results are for our strains, we put them up on the individual strain data sheets. And so this is a quick high-level snapshot of what those health reports look like. This is from one of our rooms, so AX27, you can see here. And you can find the health reports on the strain data sheets under the technical support section. But you can see that this is, again, a quick snapshot, but we have a list of the organisms we test for. We break it out into various types: viruses, bacteria, mycoplasma, etc. We tell you what the sample tested is, if it's serology, if it's tissue, if it's feces, what kind of test is done, and then what the frequency is. And then we have their most recent testing results for the past past year and a half or so. It goes for past 1, 2, 3, 4 months, and then the previous 12 months before that, so 16 months. Apologies. And then if you go into more detail, as I just said, you'll get kind of drill down into the different. We provide essentially all the testing we do, and it's very comprehensive. So if you have any questions on that, please don't hesitate to contact us in technical information. We'll be happy to help you obtain those or talk you through them in more detail. So what do you do if you find a mouse that has an infection, or if you have some kind of presentation within your facility? So if your colony has problems with infection, your best person to go to, your best resource, is going to be your veterinarian. Now, your veterinarian is going to be able to look at the specific instance and do an investigation at the time. We do get calls about this in technical support, and we're very happy to discuss them with you, but really the best person to go to, first person is with your veterinarian. So different facilities have different approaches to managing problems in immunodeficient colonies. So some facilities may opt to treat individual mice with an antibiotic, depending on how many mice are affected, what the level of the infection is, and how important that mouse is to the experiment. If it's already a mouse that's enrolled in the study, the decision to treat obviously is up to the vet and the investigator. However, if it's part of a breeding colony, it may be easier to just call the mouse and determine the extent of the infection, whether any others are involved. If it's a larger type infection, more mice are involved in that, then you may have to rederive the whole colony into the room. This may not be necessary. Again, this is where the pathogen testing comes in, where you can really start testing more and more mice and within that area to find out if it's limited to just single mouse, a single box, a single shelf, a single rack, whatever that level is. That's where your pathogen testing really comes in handy. There are, with regard to this option to treat individual mice with an antibiotic, Baytril, some antibiotics like Baytril can be effective in treating individual mice, particularly in treating mice that are part of a drug study. However, the choice to treat is up to the vet and to the investigator. Whether that might affect whether the antibiotic might affect the data coming from the mouse if it's on study is not particularly clear. Other kinds of infections like pneumocystis, which typically results in lung infections, can be controlled by treating mice with sulfatrim. It can be incorporated into feed or into the water and can result in a significant decrease in mortality. Again, we don't do prophylactic sulfatrim in our water. We acidify our water to control pneumocystis. We find that housing our mice in the maximum barrier facilities and handling them using the sterile techniques that I discussed a little bit ago is sufficient to keep our animals healthy. However, many of our researchers on the research side of JAX who are maintaining their mice in lower barrier facilities, which may be more common in many academic facilities, some of them find that administering some sulfatrim does help to keep such infections in check. And some of them use like a schedule of 1 week on, 1 week off can work. If you're interested in doing this or you'd like to discuss it further, I do recommend bringing, again, your veterinarian into this. They'll be able to make some suggestions, evaluate whether it's going to be useful, and also kind of look at it in the scope of what your testing regime is like, and develop the best plan for you, for your experiments, and for your colony. Anytime you're using these mice for humanizations, there are clearly some health considerations that I want to point out, because we are talking about health. Anytime you're working with human tissues, any human pathogens may be present, and so these samples require a BSL-2 housing and a BSL-2 laboratory. Again, anytime you're doing engraftments of any type into your mice, do testing, do a map test for mouth pathogens from any human-derived tissue. Looking for LCMV, for example. You could do a map test or culture the biomaterials, also a good way to do this. And whenever you're doing engraftments with any kind of tissue, there's a potential for graft versus host disease. This graft versus host disease is a severely when it occurs when severely immune deficient mice are attacked by human tissue or by cell transplants. So if your mice are having this kind of reaction, you can look for things like a hunched posture, ruffled fur, reduced mobility, tachypnea, which is rapid breathing, diarrhea, weight loss, hair loss, skin lesions. Time course, it happens pretty quickly often depending on the strain of mouse and also what the nature of the tissues you engrafted are. It could be days, could be weeks. You know, if radiation was involved, it may exacerbate it, and how many cells you put in. This is very common. We do engraftments with PBMCs from humans into NSGs, and obviously these develop a pretty strong graft versus host disease. You know, it's anywhere between like 6 to 10 weeks. So it's something to look for if you're unfamiliar with that. So what are some of the warning signs of a mouse or a sickness in your colony or a mouse that has an infection? It can be hard to provide an exhaustive list of what an NSG looks like when they have an infection. Non-specific clinical problems. They seem unthrifty. They have diarrhea, wasting sickness. They seem lethargic. They have a bit of weight loss, maybe that might be easier to notice. They may spontaneously have lethargy. They may die, acute or premature death, or they can, if you're breeding them, they may have some breeding problems. May have low production, small, weak, sickly pups. May have an increased rate of pup death. Smaller litters compared to the norm, or it may be dying in utero. So there's a number of different signs that may present for the NSGs, and they may not be very specific. They may be non-specific type clinical problems. And so just to drive that point home, work with your veterinarian. Again, they're first line defense for the health and well-being of your mice. So work with your veterinarian anytime you have concerns. So if your breeding performance suddenly declines, or if your mice appear unhealthy for whatever reasons, or if you're finding spontaneous deaths, or if you're concerned about some kind of testing you're getting back from your pathogen testing, your veterinarian is there. They're there to help you, and they're there to ensure the health of your colonies. And really, the better managed your colony health is, the better quality data you're getting from those mice, because you're hopefully going to be eliminating any potential confounding errors due to the infections within your immunodeficient hosts. So I hope everything we've discussed today has been helpful for you, or at least given you some food for thought. Again, remember the adage, the ounce of prevention is worth a pound of cure. It's also worth a pound of money, time, and headaches and confusion. And making sure that you understand the three main components of what constitutes your NSG or immunodeficient colonies will give you the best tools with which you can care for them and get the biggest, highest quality data that you can. They have the most impact for your research. So with that, I would like to thank you all for participating today. This webinar was made possible with your support. Jack's Mice and Services is dedicated to providing quality resources, flexible solutions, innovative technologies, and unsurpassed expertise to support and accelerate all aspects of your research. Every dollar you spend on Jack's Mice and Services supports our shared research goals and sustains valuable mouse models and resources available only from the Jackson Laboratory.